Yeah, thank you. And thank you all for coming and listening to a little bit about AlloCure. And I think I'll touch on a little of those topics that has just been mentioned. So AlloCure is a clinical stage biotechnology company, and we're focused on developing the first effective therapies for kidney disease. And our first target within this area is acute kidney injury, or AKI. And as many of you may know, AKI is a large unmet medical need. There's hundreds of thousands of patients every year who develop this condition, and currently there's nothing doctors can do. So our therapy, AC607, is an allogeneic mesenchymal stem cell therapy that's derived from bone marrow. And these cells have the ability to home to the injured kidney, where they deliver multiple factors in a paracrine manner that drives recovery and repair. So we've made tremendous clinical progress. We finished our phase 1 clinical trial that showed the therapy is safe, and we were able to generate some early efficacy data, and I'll talk about that in just a minute. We have a robust and scalable manufacturing process that we consider to be kind of a next-generation cell therapy manufacturing process, and we have a growing IP portfolio. And we have a support of investors. SV Life Science and Novo Ventures are our venture partners, and we're currently raising a B round. So we have an experienced management team led by Dr. Robert Brenner, who's a nephrologist and has had years of experience in Amgen and AMAG. So just a little bit more about AKI. As I mentioned before, AKI is a completely unmet medical need. If patients develop this condition, there's nothing doctors can really do, and there are many risk factors that predispose patients for developing AKI, and they're very common: chronic kidney disease, diabetes, hypertension, even old age. And what often pushes patients into an AKI setting is when they go in for cardiac surgery or when they have contrast imaging agents or nephrotoxic drugs. And once this condition sets in, the consequences are quite severe: lengthened hospital stay, onset or progression of chronic kidney disease to end-stage renal disease, dialysis, of course, and oftentimes death. So the properties of AC607, as I mentioned, these cells are derived from bone marrow, from healthy bone marrow donation. And from that, we're able to manufacture a unique population of mesenchymal stem cells. Our therapy is not transformed or differentiated, so it's genetically stable. And importantly, it's not recognized by the immune system. So we don't have to worry about tissue typing or donor matching. It's an off-the-shelf product that can be shipped out and used by hospitals. Our therapy acts through paracrine mechanisms, and it does not persist in the body. And generally, MSCs are considered to be quite safe and have a high safety profile. So in many ways, our product is like a standard specialty pharma product that will fit into standard sales and distribution models that are used today. So a little bit about the mechanisms. AC607 really has an elegant mechanism of action because it is amplifying or magnifying an endogenous injury response. When the kidney is injured in an acute setting, it gives off a chemokine called stromal-derived factor 1. And this SDF1 draws MSCs out of the kidney, where they then home to the damaged kidney. So with our therapy, we deliver our therapy. The therapy homes to the damaged kidney through the same SDF1 response mechanisms.
Draws them out of the marrow.
Yeah, it draws them out of the marrow. Did I say the kidney? I'm sorry. Draws them out of the marrow, where they're taken right out into the kidney. And once they're in the kidney, there's a multifactorial response. It's anti-apoptotic, mitogenic, anti-inflammatory, and angiogenic. And this polypharmaceutical approach, delivered by our cells into the kidney, really is what drives the recovery and the repair and reverses the course of this very serious disease. So our manufacturing process is what we would consider a next-generation manufacturing process. It's a completely closed system. It's free from animal-derived products, and it's easily scalable to meet our phase 3 and then commercial scale with attractive cost of goods sold. We package it in cryovials. It can be shipped out, stored at the hospital, and then delivered to any patient who needs the therapy. So our phase 1 trial summary, we delivered these cells to cardiac surgery subjects who are at high risk for developing acute kidney injury after their surgery. And the results showed that the therapy is, of course, safe, has a very high safety profile. And we were able to use a historical matched case control from the same institution that we did the phase 1 trial. And those results, when you compared the two, were quite striking. The incidence of AKI was lower in our treated subjects, which is represented in the blue bars. And this was done using two different diagnostic criteria, Rifle and Aiken. In addition, the length of hospital stay and the readmission rates were also lower. Now, while these data are early, it's very encouraging and has certainly given us a lot of confidence as we move forward into our phase 2 trial, which will be a large, multi-centered trial done at leading institutions across the country. And we were just awarded Fast Track status by the FDA. So to wrap things up here, again, this is a large market, and we did a commercial analysis where we presented the product profile of AC607 to payers and doctors, and there was almost universal agreement about the tremendous health and economic benefits this could have to patients. So I look forward to taking some questions.
Or questions. You presented phase 1 data. Can you tell me a little bit about what the design
Would you tell me a little bit about what the design criteria was for the phase 1 clinical trial? What was the entry criteria for these patients? Was it blinded, placebo-controlled? How did you do that? What gives you confidence that those p-values are real that we just saw? And especially small trials, you don't normally see p-values with those kinds of small numbers.
Yeah, good question. So the criteria for our trial was really, it's a standard phase 1 trial. It wasn't placebo-controlled or blinded, but it was done at an institution that had this wonderful database that we could mine to get these data. So it was a standard trial, patients that met high-risk criteria for developing acute kidney injury. These are patients that came in, and most of our patients had chronic kidney disease. Many of them had diabetes, and the average age of our patients was 75. So they were really primed for AKI events post-surgery.
And did you explore a dose response in this trial?
We did. We did 3 different doses.
And what did you find with those?
And we found that, that in the different dosing levels, we found that there was some minor differences, and this has really informed us in that, along with our preclinical data, on what dose we're going to move forward with in the phase 2.
Yeah, what I was really pushing for was, did you find a biologic threshold at which it didn't matter when you went up in dose? Was there a critical mark?
There was, the lowest dose certainly was not as effective. We saw that the middle and the high dose definitely were better at some of these criteria we were looking at.
And the end results were, what would be an endpoint in a phase 2 clinical trial? Would it be a qualitative marker like creatinine or Boutin, or would it be, something more relative like hospital stays or, days on dialysis?
Yeah, that's a very good question. The FDA these biomarker, like even creatinine, is a difficult endpoint for phase 2 pivotal type trials or phase 3 trials. So really what we're looking at in the phase 2 is going to be time to kidney recovery, and then we'll also look at death and dialysis.
Okay, terrific. Thank you so much.